In this lecture, we're going to be talking about plant cells, and you should read Chapter 3 in your textbook. I also want to remind you that in class on Thursday, we'll actually be focusing in on some of the major plant cells cells that you find specifically in plants. Okay, so first off, I wanted to talk a little bit about the history of cell theory and our ability to even be able to see cells. So Robert Hooke used the term cell for the discrete structure he saw when he looked at a thin slice of cork with his microscope in 1665. So this little inset here is basically what he saw. In the 1800s, 3 scientists made observations leading to the development of cell theory. And basically, this theory states 5 things. 1, that all organisms are made up of cells. 2, new cells are always produced from pre-existing cells. 3, the cell is the structural and functional unit of all living things. 4, the cell contains hereditary information which is passed on from cell to cell during cell division. And lastly, that all cells are basically the same in chemical composition and metabolic activities. Cell theory is a major scientific achievement and is the basis for many advances in modern biology, including controversial issues such as cloning and cultivation of human stem cells. So when we talk about organisms, we can classify them as unicellular or multicellular. As the name suggests, unicellular would be composed of just one cell. So that includes things like bacteria and archaea, many protists, and some fungi. So when we will be talking about protists, we will talk a little bit about some organisms that are unicellular, but for the most part in this class, we'll be dealing with multicellular organisms. Organisms that are composed of more than one cell. So most seaweeds, fungi, plants, and animals are all multicellular. So we can classify 2 different types of cells, prokaryotic versus eukaryotic. And once again, we aren't going to spend too much on prokaryotic cells. They're the cells that lack nuclei and other membrane-bound organelles are found in archaea and bacteria. But we're going to be focusing on eukaryotic cells, so the cells that possess a nucleus and other membrane-bound organelles. However, there are 3 components within cells that are found in both eukaryotic and prokaryotic cells. So these things are a cell membrane, which encloses the cell contents, a cytoplasm, which is the watery solution that includes other cell structures, and ribosomes, which is composed of protein and RNA, and this is where they synthesize proteins. So I'm not going to go into a lot of the cell features that are common in eukaryotic cells, but I do want to talk about cell membranes. And the reason I want to talk about that is this whole idea of a fluid mosaic model and a phospholipid bilayer. It's really important when we're talking about cell communication, the ability to transport materials, and it's very important when we talk about photosynthesis. So I want to make sure that you have an understanding of this before we move on. So the cell membrane, once again, encloses the cell contents, and it's composed of phospholipids, proteins, and other materials. And the fluid mosaic model is the current model for the structure of the plasma membrane and many other cell membranes. And this basically is that protein molecules will float in a phospholipid bilayer. Now, the phospholipid bilayer is composed of hydrophobic and hydrophilic heads. And the best way of basically understanding that is to look at a cartoon representation. So here's our cartoon representation of a membrane structure. So you can see these proteins that are embedded, and then you can see that there are these 2 layers. So we've got a layer here on top and then a layer below. And what I want you to notice is that you have the hydrophilic heads are going to point outwards towards the water. So let's say this is a watery substance, and you have the the hydrophilic heads are pointing into the water, and the hydrophobic tails point inwards so that they're not exposed to the water. Once again, notice that there are proteins embedded into this, and this is important because this kind of structure allows for materials to pass through, and can set up a concentration gradient that's really important in photosynthesis. So understanding this kind of phospholipid membrane structure is important for later lectures. So once again, I really want to focus just on some cells that are really important in plants, but I just want to mention that all plant and animal cells do have a plasma membrane. They have mitochondria and a nucleus, ribosomes, an ER, the Golgi apparatus, and the cytoskeleton. Most of you have had 121, so you're really familiar with what these things are, and it's also in your textbook. But once again, for testing and for activities and things, we're really going to focus in on the characteristics that are found just in plants. Okay, so the first thing that I want to talk about is the cell wall. So the cell wall is comparatively rigid. It supports the exterior wall to the plasma membrane in plants, also fungi, prokaryotes, and certain protists. It protects plant cells from osmotic stress and also protects them from disease-causing microbes. And it contributes to the plant body shape and its strength. So the components of the cell wall is cellulose. It's a major structural component. It's composed of long cables containing cellulose fibrils that are bound together by other components, and it can constitute about 30% of the dry mass of plants is the cellulose. So here in this diagram, it is highlighted as these green tubes. Non-cellulose components are the hemicellulose. It binds the cellulose fibrils together. So here you can see that as these little orange lengths. And then pectin, it combines with calcium and water, and it makes kind of a jelly-like matrix between the cells. And then also proteins. And so you put that all together, and it helps to form that rigidity within the cell wall of plants. Okay, so I also want to talk a little bit about the differences between primary versus secondary cell walls. So when we talk about primary, and here you can see that as this green layer right here. This is a single layer cell wall of growing cells that maintain the ability to stretch. When we talk about secondary, these are laid down inside the primary wall as cells mature. And this is where lignin may be added to the walls of these water-conducting cells, and it makes them impermeable to water. So this second wall, it's thickening it, and the lignin is also kind of a protective action. So here you can see this in more detail. If you look here, you can see this primary cell wall and then the thickenings that are occurring with that lignin. So I don't know if the term lignification is something that you are familiar with, but you probably have noticed young seedlings or plants where their first year they're often green. So think about like an oak seedling. So the first year after it germinates from a seed, it's got a very green stem and it's very flexible. And then over time in the next summer, it starts to get a much harder stem, and we call that lignification. And it basically is that you're getting the secondary wall thickening, and it's getting woodier and more rigid, basically. So it's a thickening and more rigidity of that wall by the addition of lignin. And that lignin is important because, as I mentioned, it makes the cell wall more impermeable to water, and it also is protective. So it protects the plant from a lot of different pathogens that can kill the plants as well. Okay, another thing that are found in plant cells only is plasmodesmata. So these are channels that run through cell walls connecting the cytoplasm of adjacent cells. And the channels are lined with cell membranes, and they contain strands of ER. And so these are very important as well. They allow for the transfer of materials and communication between cell to cell. Okay, the next thing that's really critical is the chloroplast. This is important because this is the site of photosynthesis. So leaves and other green parts of the plant will contain these organelles, and once again, the chloroplasts contain chlorophyll, and this is an important component for the ability to photosynthesize. So cells can have between 40 to 50 chloroplasts. They're composed of thylakoids, which are arranged in stacks, and they are also a subset of plastids. So we'll be actually looking at a number of different plastids, and these are a group of membrane-bound organelles that occur in the photosynthetic eukaryotic cells. Some of the other type of plastids that we'll be looking at are the amioplasts. These are non-green plastids that store starch. So for example, we'll be looking at potatoes, and you probably have heard never to eat a green potato, or that you shouldn't eat a green potato. The potato turns green when it is in the light, and that's because the amioplasts, which store the starch, actually turn into chloroplasts when exposed with light. So these plastids can go from one state to another depending upon environmental cues. Another plastid is chromoplasts, and these contain carotenoid pigments that color leaves, flowers, and fruits, and so they can color things red, orange, or yellow. And so these chromoplasts are important for a variety of reasons, and we'll talk a little bit more about their function later. But many of these pigments are important in photosynthesis, and they can be also important as deterrents for herbivory. Also, what's found in plants are vacuoles. So these are a large fluid-filled membrane-bound sac within the cytoplasm, and it contains a solution of salts, ions, pigments, and waste materials. Vacuoles may occupy about 90% of the cell volume, and in this figure you can see a pigmented vacuole of a stamina hair of spiderwort, and it dramatically conveys the size of the vacuole. So pretty much this entire thing is composed of a vacuole. The nucleus and cytoplasm are restricted to the very edge of the cell. And what is the importance of the vacuole? Well, it provides turgor pressure for the cell, so it helps to maintain plant structure. Also, if the plant cell becomes and this is important, or one way of looking at this is if a plant cell becomes dehydrated, the vacuole loses its water, and eventually the plant will wilt. So you can see its function very readily within that ability for it to maintain its structure and to be upright. That really has to do with having this turgor pressure because of the vacuoles. It can also store proteins, carbohydrates, pigments, tannins, and it can store heavy metals. So it's used for a variety of things, anywhere from the ability to help to change osmotic potentials so that plants can get more water out of the soil. It can be used to take up toxins in the soil, a place for pigments or tannins to be stored in order to decrease herbivory. So all of these things can happen or be within a vacuole, and so it has many different functions. Also, this is the site where the breakdown of proteins and other complex molecules can occur that are no longer needed by the cell. I do want to mention, we'll talk about this a little bit later, but the ability for plants to have these vacuoles is really important for reclamation and restoration. A number of plants are able to grow in soils that are rich in heavy metals, for example, near old mines or military bases, because they have the ability to hyperaccumulate these toxic metals. So the metals are combined with proteins and stored out of the way in the vacuole. This may make the plant toxic, and it also definitely deters herbivores. And these plants are really useful in cleaning up contaminated soils. So we'll talk a little bit about phytoremediation later in the semester. Some non-plant cells may contain vacuoles, but they're generally much smaller than those of plant cells. Okay, so the next two slides are just a table that gives you some of these different structures. Some are found in all types of cells, and others are just found in both animals and plants, and then others are just found in plants. So it gives you the structure, the description, and then the function. And so I just thought that this would be a useful way of very quickly organizing the information as you're looking through it and studying for your test. So just as a reminder, we will be meeting in 1:15 on Thursday, and we'll be doing microscope work looking at various components of these characteristics of cells that are found in plants.